everyone, I am your BSc student, Michaela BILLONES. And for today's video, I will be discussing about the process of mitosis and meiosis. What is mitosis? Mitosis is the process in which eukaryotic cell nucleus splits in two, followed by division of the parent cell into two daughter cells. The word mitosis means threads, and it refers to the thread-like appearance of chromosomes as the cell prepares to divide. Phases of mitosis. Mitosis consists of four basic phases: the prophase, metaphase, anaphase, and telophase. Prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus known as chromatin condenses. Metaphase. It is a stage in the cell cycle where all genetic material is condensing into chromosomes. These chromosomes then become visible. During this stage, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell. During this stage, in human cells, the chromosome then become visible under the microscope. As metaphase continues, the cells partition into the two daughter cells. Anaphase is the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Before anaphase begins, the replicated chromosomes, called sister chromatids, are aligned along the equator of the cell on the equatorial plane. The sister chromatids are pairs of identical copies of DNA joined at a point called the centromere. During anaphase, each pair of chromosomes is separated into two identical, independent chromosomes. Telophase. It is a process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell. During telophase, a nuclear membrane forms around each set of chromosomes. To separate the nuclear DNA from cytoplasm, along with telophase, the cell undergoes a process called cytokinesis that divides the cytoplasm of parental cell into two daughter cells. Cytokinesis. It is a physical process of cell which divides the cytoplasm of a parental cell into two daughter cells. It occurs concurrently with two types of nuclear division called mitosis and meiosis, which occurs in animal cells. Mitosis, each of the two meiotic divisions result in two separate nuclei contained within a single cell. Cytokinesis performs an essential process to separate the cell in half and ensure that one nucleus ends up in each daughter cells. Meiosis. Meiosis is a type of cell division that produces the number of chromosomes in the parent cell by half and produces four gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring. Prophase 1 in meiosis. The first meiotic division begins with prophase 1. During prophase 1, the complex of DNA and protein known as chromatin condenses to form chromosomes. The pairs of replicated chromosomes are known as sister chromatids, and they remain joined at the central point called the centromere. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side or pole of the cell. Metaphase One. Between Prophase One and Metaphase One, the pairs of homologous chromosomes form tetrads, which the tetrad any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination. Recombination is a process that breaks, recombines, and rejoins sections of DNA to produce new combinations of genes. In metaphase 1, the homologous pairs of chromosomes align on either side of the equatorial plate. Anaphase 1 is the spindle fibers contract and pull the homologous pairs, each with two chromatids, away from each other and toward each pole of the cell. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. Each daughter cell is haploid and has only one set of chromosomes, or half the total of chromosomes of the original cell. Prophase 2 and Meiosis 2 is a mitotic division of each the haploid cell produced in Meiosis 1. During Prophase 2, the chromosomes condense and a new set of spindle fibers form, and the chromosome begin moving toward the equator of the cell. Metaphase 2 and Anaphase 2. During Metaphase 2, the centromeres of the paired chromatids align along the equatorial plate in both cells. Then, in Anaphase 2, the chromosomes separate at the centromeres. The spindle fibers pull the separated chromosomes toward each pole of the cell. Lastly, the telophase 2 is the chromosomes are enclosed in nuclear membranes. Cytokinesis follows, dividing the cytoplasm of the two cells. At the conclusion of meiosis, there are four haploid daughter cells that go on to develop into either a sperm or egg cell. Before I end this topic, I will leave a quotation saying, "Whenever you feel sad, just remember that there are billions of cells in your body and all they care about is you." Thank you for watching.